Hi, my name is Melisa and I'm a graduate student in Koç University. Today I will talk about how protein-protein interaction networks reveal crosstalk proteins and pathways related to vascular cognitive impairment. Vascular cognitive impairment, or VCI, is referred to as a heterogeneous condition because it is cognitive impairment caused by vascular factors. It is the second most common type of dementia, and it is generally a problem seen in older adults, and the aging population has increased in the past century. It is important to study VCI because it causes memory and attention impairment, and also decline in executive functions and information processing speed. And these characteristics make it crucial to understand vascular cognitive impairment. By researching VCI, we can identify potential biomarkers for early diagnosis and detection. We can also identify therapeutic targets, and we can learn more about the complex relationship between vascular and cognitive health. But as vascular cognitive impairment is a complex disease that contains both cardiovascular and cognitive components, and also because their precise relationship is not known, it is hard to study it experimentally. In this study, we aim to find proteins and pathways associated to vascular cognitive impairment by studying the crosstalk between cardiovascular and cognitive diseases. We use protein-protein interaction networks to study this crosstalk. We built protein-protein interaction networks related to 5 cardiovascular diseases and 2 cognitive diseases. We also included the protein-protein interaction network of oxidative stress, because it is a known factor related to VCI, cardiovascular diseases and cognitive diseases. And we also regard oxidative stress as a possible mechanism related to crosstalk. So for each of these 7 diseases and oxidative stress, we built a protein-protein interaction network. We use Gildify web server for this. This web server builds PPI related to the disease by Gildify association principle, and it provides a score ranging from 0 to 1 for each protein in the network. To study the crosstalk, we merged the 8 PPI networks and we called it the global network. We next found the proteins that are both central in the network and are related to oxidative stress network. Here you can see the top 5 proteins for all the oxidative stress related proteins. We next wanted to find which of these proteins are more affected by oxidative stress. So we utilized the scores given by our tool and we created 2 scores with these. An oxidative stress included score and an oxidative stress excluded score. We checked the difference between these scores and saw that it ranges from positive to negative 0.2. We selected 0.1 as a threshold such that if the difference between these scores is at least 0.1 or less than negative 0.1, oxidative stress has an important effect. By doing this, we found the central oxidative stress proteins. And there were 12 proteins that were central to the network, and the effect of oxidative stress is at least positive or negative 0.1. Next, to find which pathways these proteins can correspond to, we performed pathway enrichment. We selected the cardiovascular interactors of these 12 central oxidative stress proteins, and we enriched them. This resulted in the HRH pathway. And also, we enriched these 12 oxidative stress proteins with their cognitive disease interactors from the global network. And we ended up with the notch signaling pathway. We later investigated if a crosstalk between these 2 pathways was previously proposed. And we saw that the crosstalk of these 2 pathways was seen in other systems. And also, previous research showed that the notch signaling pathway may cause mesenchymal transition, but its relation to the blood-brain barrier with this path was not shown. Therefore, we hypothesize that the crosstalk between age-range pathway and the notch signaling pathway may be causing mesenchymal transition, leading to blood-brain barrier damage and leakage, causing increased permeability. By the loss of selectivity in the blood-brain barrier, oxidative stress will increase, and the increased oxidative stress levels can increase the amyloid beta production in the brain. And this mechanism as a whole can be causing vascular-originated cognitive degeneration. And lastly, MG, which can cause protein glycation by post-translational modifications of particular amino acids, was used to test the relevance of oxidative glycative stress in vitro. According to cell viability assays and caspase-3 activation, we discovered that 250 micromolar Mg toxically affects mature murine cortical neurons in a statistically significant way. So as a result, we proposed a crosstalk pathway for vascular cognitive impairment by studying protein-protein interaction networks of cardiovascular and cognitive diseases. And this crosstalk of pathways is between the age-range and the notch signaling pathways. And this crosstalk can be disrupting the blood-brain barrier via the endothelial or epithelial to mesenchymal transition. Here are our references and acknowledgements. Thank you a lot for your attention.